This is a lab video from Science Bridge in high-tech high. Enzyme substrate, get a reaction.
Have you ever wondered why apples turn brown after leaving them cut open for too long? This is the day you will get to hands-on understand how enzymes work and how variables affect them. Today you will be examining the enzyme responsible for turning mushrooms and also apples brown, the tyrosinase enzyme. You will be grinding a portobello mushroom to extract this enzyme from it. You will then introduce variables that will affect the reactions. First, you need to get your mushroom piece, put it in the weighing boat, and put 4 milliliters of grinding buffer on top. Grind up your mushrooms with the grinding buffer until they are mixed up well. There's going to be some liquid after you do this. Pour it through a filter into a test tube. This liquid is your tyrosinase enzyme, the enzyme you'll be observing in this lab. Now, make sure that everyone in your group is ready to jump on it. Make sure that your roles are assigned before you start the process. The reaction you will do next is going to move very fast and will start right away. You won't have time to get organized at that point. Be ready with your data chart, timer, and calorimetric chart. If you're wondering what that is, it's a chart that measures the amount of product, in this case pigment, being produced by the tyrosinase and L-DOPA reaction. Before you move forward, make sure you know how to properly use a pipette. In all of your trials, you will need to use one. A pipette is extremely handy and is used to transfer liquids. To use a pipette properly, you first need to squeeze the top part of it, which looks like a bulb. Next, place the tip of it into the liquid you want to transfer, and slowly release without fully letting go. You don't want liquid in the bulb. By doing this, the pipette will suck up the liquid into itself. Finally, you move the pipette to where you want to place the liquid, and slowly squeeze the bulb again so that it may release the liquid. The first trial is a control trial, meaning you will observe the activity of the enzyme under standard conditions. You will put 2 milliliters of L-DOPA into a different test tube labeled control. L-DOPA works as a substrate. A substrate is a molecule that an enzyme acts upon. Remember, when the substrate reacts with the enzyme, its bonds are weakened and the substrate changes shape. You will then add 0.1 milliliters of the tyrosinase enzyme using a pipette to the test tube with the L-DOPA. Swish it around constantly so you'll be more likely to see all the colors. When you add the tyrosinase enzyme, immediately start to record the color change on your calorimetric chart. Do this for 7 whole minutes at time intervals. The tyrosinase enzyme catalyzes the reaction and converts the L-DOPA to dopachrome, which is a reddish-brown color. Empty the test tube. You are ready to move on. Next, you will be conducting one experimental trial. This means you'll be repeating the process, but with other variables added in. You can choose one of these trials: pH, temperature, or the use of an inhibitor. For pH, begin with putting 2 milliliters of L-DOPA into a test tube. Take 1 drop of that and place it on pH paper. If you're wondering what pH is, it is a measure of acidity or alkalinity. Determine the pH and record it down. Next, transfer everything from your acid tube into your EXP tube. Record your pH once again. Make sure to remember that you need to record the pH twice, once before you add the acid and once after. Next, add 0.1 milliliters of your tyrosinase enzyme to the EXP tube. Start timing immediately and use your calorimetric chart to record the color at every time interval. Dispose of your materials safely, and you're done. Through this experiment, you were able to see the effects of pH on the enzyme. If you would like to choose the temperature trial, add 0.1 milliliters of tyrosinase to the EXP tube. Place the tube in 90 degrees Celsius water for 1 minute. After this, add 2 milliliters of L-DOPA to the tube and record your color change at time intervals. Make sure you add all of the L-DOPA at once, and start recording immediately. By adding sodium benzoate, you can observe the effects of an inhibitor on the enzyme. Begin with adding 2 milliliters of the L-DOPA to your EXP test tube. Then, add all of the contents of the inhibitor tube to the EXP tube. I'm sure you realize what's coming next. Record your color change at time intervals. Then, add 0.1 milliliters to the tube and continuously mix. Record your color changes, but remember to mix the entire time. Congratulations! You're done with each trial. Remember to dispose of your materials. Next, you will be analyzing your results. Today, you will follow these steps to learn about enzymes and enzymatic reactions. From taking an easily extracted enzyme from portobello mushrooms, you can observe these reactions using different variables to learn about the functions of enzymes and how they work.